Hello everyone, welcome to Molecura. Today we're going to talk about enzymes and how do they catalyze chemical reactions. These concepts are going to be very important for better understanding medical biochemistry and metabolism and its reactions. So enzymes are macromolecules that act as a catalyst to speed up a chemical reaction. Now, let's say you have 1 mole of glucose and you have it sitting in your kitchen floor. So this 1 mole of glucose, if you leave it there for 6 years, it's not going to be turned into energy in your kitchen. But the same 1 mole of glucose, if you put it inside a cell in your body, is going to be turned into energy in less than a minute. So what is the difference between your kitchen floor and inside a cell? What's going to make this reaction happen at a much higher rate than outside the body? And your answer is going to be enzymes. Presence of enzymes is going to make this reaction become much faster. So enzymes are acting as a catalyst to speed up a chemical reaction. Now, most of the enzymes are made of proteins, but not all of them. We know that some enzymes could have a form of nucleic acid, or namely RNA. But how are the enzymes speeding up this chemical reaction? Enzymes are going to be speeding up the chemical reaction by lowering the activation energy. What do we mean by that? Before we go into the graphs, let's try to look at an example. So our example here is going to be a mountain. So we have a mountain here that we are going to take a very heavy ball up this mountain and we are going to take it to the other side. So the way to do it is you need to use a lot of energy to take this ball first up the mountain. And once you do this, all you need to do is to throw this ball and it will roll down and go to the other side. So the first step is going to be very important because this ball is quite heavy and you need to carry it all the way up this mountain. And once you get the ball here, it's just going to roll down to the other side. This part does not require much energy. So the main energy is taking this heavy ball from this point to this point. And you would need to spend a lot of energy to do that. So what are the enzymes doing? The enzymes are going to come here and they are going to help you to take this ball much easier to the top of the mountain. And the amount of energy that you need to use here to take this ball up the mountain is called the activation energy. So the enzymes are doing the same thing with chemical reactions. Here we have the substrate and they want to take this substrate and turn it into products. Before they do that, they need to reach the transitional state, which is here. So they would need to apply energy, take this heavy ball up the mountain and then roll it down. The exact same thing is happening when we are talking about chemical reaction, and we're going to show it on the graph. Here, the reaction is going to happen without enzymes. So as you can see on the graph, our y-axis is energy, same as here. Now we are having the energy level of the substrate, plus we have the energy level of the product. So this is our substrate and this is our product. Now, for this reaction to happen, we are going to need to take it first to the transitional state, which is here. So the energy level needs to rise, go up to this point, then come down and reach to the product. So the amount of energy that we are using to reach this substrate to the transitional state is called the activation energy. Let's try to write it down as well. So this is called activation energy. Activation energy again is the amount of energy needed to take the substrate to the transitional state, and from there on it's going to be an easy ride to make the product. Now this reaction is happening without enzymes and we are going to need a lot of energy to make this reaction happen. But when enzymes are involved, this is going to be much easier and much faster. So here we are going to have our enzymes involved. So what is happening again? The same thing. We are going to have our substrate. We are going to have our product. But now the transition state is much lower than it was before because the enzymes are lowering the activation energy. So this reaction is going to happen at a much faster rate. And as you can see here, there is a clear difference between the two. So this is our activation energy. This is our activation energy. Now, as you can see, what has happened here is that the enzyme has taken this activation energy, which was a large amount, and has made it into a smaller activation energy. So this is how the enzymes are going to catalyze a reaction and are going to make the reaction happen at a much faster rate compared to situations where enzymes are not available. So when a substrate is going to get converted into a product with the presence of enzymes, this reaction is going to happen fast and it's going to happen more often. So now let's try to look at the structure of an enzyme. So let's say this is our enzyme, and what we are having here is an active site. Active site is the site where the enzyme and the substrate bind to each other. So this is our active site. And as we can see, our substrate has a similar shape and can easily find its way into the active site. So here in this picture, this is our substrate, and this here is our active site. This is quite important because some medications and some chemicals, some toxins can bind to an enzyme to the active site, and they can try to disturb the binding of substrate to the active site of the enzyme, therefore stopping that chemical reaction from happen. But what we need to understand from this slide is that the enzyme and the substrate have a complementary shape, which is going to make the substrate bind easily to the enzyme and for the chemical reaction to happen. Most of the time, the enzyme itself remains unchanged. So once this substrate gets detached from the enzyme would be ready for the next reaction to happen. Now, for some enzymes to function, they're going to need cofactors, and we're going to be discussing this in more detail when we are discussing specific enzymes in metabolism. Some of them are going to need inorganic molecules like zinc and copper, and some of them are going to need organic molecules like vitamins. When cofactors are made of organic molecules, we are going to be calling them coenzymes. So coenzymes are organic molecules which are acting as a cofactor for enzymes, and this could be vitamins, for example, vitamin B1, B6, and so on. Now, the next topic that we need to discuss briefly is going to be enzyme inhibition. Enzyme inhibition could be three types in general. The first one is competitive inhibitor, which there is a weak bond between that inhibitor and the active site of the enzyme. So what is happening here is that we are going to have our enzyme. This is our enzyme. Again, this is our active site. And we have the usual substrate that binds this place, but we are going to have a competitive inhibitor This is our competitive inhibitor, which is going to inhibit this enzymes by binding with a weak bond to the active site. Now, coming to the second point is non-competitive inhibitor. This is going to be usually irreversible, and what we have in non-competitive inhibitors is formation of covalent bond, changing the enzyme's structure overall. So the covalent bond that gets attached to the enzyme is going to change the structure of the enzyme altogether. So what is happening here is that we are having this as a non-competitive inhibitor. We're going to have a non-competitive inhibitor come here and form a covalent bond with our enzyme. So what is happening is now that the structure of this enzyme is getting completely changed. So there will be no more attachment of substrate and this enzyme because the structure has completely been distorted. This is what we call a non-competitive inhibitor. Now, uncompetitive inhibitor is going to be when inhibitor binds to enzyme substrate complex, stopping the reaction from happening. So what happens in uncompetitive inhibitor is that you already have your enzyme and you already have your active site, and your active site is already occupied by your substrate. This is your substrate. But an uncompetitive inhibitor is going to come and it's going to get attached to this complex, and it's going to prevent the substrate from detaching from the enzyme, practically making this enzyme and this substrate useless for the cell. So this is called an uncompetitive inhibitor. We are going to talk more about in a separate video about what these competitors mean, like by reducing the speed of the reaction and by requiring more concentration of substrate for the reaction to go forward, and we will talk about those things in detail. For now, we're going to be understanding what competitive inhibitor is, what non-competitive inhibitor is, and what uncompetitive inhibitor is, because to understand the chemical reactions that are happening in glycolysis, in TCA cycle, in beta oxidation, or in electron transport chain, you would need to have a basic understanding of the enzymes. Now we will talk about enzyme regulations. The enzymes in our body are heavily regulated, which means that the enzymes are going to work when the cell needs them to work, and they are going to stop when the cell does not need them anymore. So the first thing that we're going to talk about is going to be allosteric regulation. Allosteric regulation is going to be, again, we have our enzyme. This is our enzyme. We have our active site here. And our active site is going to have substrate coming to get attached to it. Now, on completely other side of the enzymes, there is a binding site. I'm just drawing it like this for the sake of simplicity. We have another molecule comes and gets attached to this allosteric site and is going to regulate the reaction that is going to happen here. So to try to name this is our allosteric binding site. This is the allosteric binding site, and we are going to have some molecule get attached to this, and this molecule at the end is going to affect what is going to happen to this substrate. It either is going to increase the rate of the attachment of substrate to the active site, or it is going to inhibit it from binding to the active site. So this is what we call an allosteric inhibition. Now we're going to be talking about allosteric inhibition a lot when we are talking about glycolysis and the TCA cycle. So you should have an understanding that one enzyme has an active site, and this active site binds to a substrate. But there are other sites on the enzyme as well, which are called the allosteric binding site. What happens when one molecule that allosterically regulates this enzyme gets attached to this area, and this attachment is going to send feedback to the substrate and to the active site to either tell them to go ahead with the reaction or to tell them to stop the reaction where it is. Another type of enzyme regulation is going to be feedback inhibition. Feedback inhibition is going to be much more simple. It is when product of an enzyme inhibits the reaction that's catalyzed by that enzyme. Let's take an example of glucose in the first step of glycolysis. Glucose gets converted into glucose 6-phosphate. And this happens through the action of an enzyme that we call hexokinase. Hexokinase gets regulated by the product that it's making. So if this reaction happens a lot, we're going to have a lot of glucose 6-phosphate. And the glucose 6-phosphate is going to come here and is going to tell hexokinase to stop this reaction. We don't need you anymore. You need to stop this reaction. It's going to negatively regulate hexokinase. So as we can see here, the product of this reaction is inhibiting this reaction from happening over and over again because we already have enough glucose 6-phosphate and this does not need to go ahead. Other types of enzyme regulations which we are going to talk about when we are going through metabolism. So the first one that we're going to talk about is gene expression. As we know, enzymes are made of proteins, and inside the cell you're going to have, this is our cell, you're going to have a nucleus. Your nucleus is going to have DNA in it. And for some enzyme to start working, you would need this to be transcribed into mRNA. And you need this mRNA to get translated into a polypeptide chain. And this polypeptide chain at the end is going to become an enzyme, which is a protein. So sometimes what happens is that we need a lot of this enzyme. And there are molecules or hormones that are going to come and they are going to tell this DNA that we need you to activate these genes. We need more transcription of this mRNA to happen because we need more translation of this mRNA into polypeptide and we're going to need more of these enzymes. So this is how gene expression is going to work on regulating enzyme activities. The second thing is going to be substrate availability. The rate of the activity of the enzyme is dependent if the substrate is readily available or not. So if the enzyme cannot find any substrate available in the cell to do the reaction, obviously the activity is going to stop. The third thing that we're going to talk about is going to be compartmentalization. So what does this mean is that the cell has many compartments. The enzymes and the substrates are going to be kept in a specific cellular compartments unless they need this reaction to go ahead. For example, some enzymes could be in the mitochondria, some enzymes could be stored in the vesicles, in the lysosomes, in the Golgi apparatus, and so on. So the compartments of the cell are going to affect if the substrate is readily available to the enzymes or no. And this is also going to affect the activity of the enzymes. Again, these points we are going to be discussing them in more detail as we see examples when we are going forward with the reactions that happen in metabolism. With this, we end our lecture on enzymes and their activities. Next, we are going to be talking about the overview of oxidative phosphorylation, how our body makes energy, and then we will look into glycolysis and the TCA cycle. Thank you for watching. If you enjoyed this content, make sure to subscribe to our channel so you could explore more videos on Molecura.